Hi, Tony Voyers, CEO of Advanced Chemotherapy Technologies. I'm joined today with our CTO, Bill Daunch, and one of our founders and clinical advisors, Dr. Jinjin Ye. We appreciate the opportunity to talk to you about our approach using iontophoresis for drug delivery in very challenging tumor types, namely pancreatic cancer. We've developed a scalable clinical stage drug delivery platform technology that we're moving into our first in human clinical trials later in 2020. With this technology, we deliver high doses of chemotherapies directly to the tumor tissue with significantly lower systemic levels, potentially reducing systemic toxicity. Additionally, we have the ability to deliver multiple drugs at the same time in drug combinations, permitting a high level of customization based on patients' needs.
Let me step back and tell you why we are so excited to bring the device to pancreatic cancer first. Over the last decade, there has been a doubling of pancreatic cancer cases in the U.S. and worldwide. While we have seen treatment advances during this time, 5-year survival is still 9%. Clearly, we need to do better. Similar to many other tumors, surgery provides the best chance of cure. But only 15% of patients who get diagnosed with pancreatic cancer will be able to undergo surgery. In fact, almost half of patients who might be eligible for surgery cannot have it because the tumor is wrapped around critical structures like the superior mesenteric artery and vein seen in the picture. If we could just shrink the tumors enough to get them away from these structures, surgeons could go in and take the tumor out and give the patients a chance for cure. These are the patients that we are trying to help.
Shown in this slide is a schematic of our iontophoresis drug delivery system. First, our drug delivery electrode is implanted onto the patient's pancreas near the tumor. After recovery from the surgical procedure, the patient returns to the hospital infusion center to begin the chemotherapy. At that time, a counter electrode is applied to the patient's back, and our custom DC power control is attached to the counter electrode and to the electrical port. Standard infusion equipment is attached to the port with the chemotherapy lines, and then the power is turned on where the chemotherapy is delivered directly to the tumor by the electrode. A typical course of treatment would be weekly infusions for 8 weeks. When the treatment is over, the tumor would be re-imaged, and if the tumor is shrunk away from those blood vessels Jinjin mentioned, both the tumor and the device would be removed.
I'd like to present some of the exciting preclinical data that brought us to this point today and was published in Science Translational Medicine. For our studies, we used patient tumors that were implanted into the mouse pancreas because this is a good model to predict response in patients. We compared device-delivered gemcitabine with placebo or standard IV delivery. On the graph is plotted the change in tumor growth over the study period. In black is the no treatment group. In orange, you see the tumor growth with IV or systemic delivery. While the tumors slowed down, there was still a 140% increase in size. What was amazing to us, and continues to be amazing as these tumors are very refractory to treatment, is the shrinkage of 40% with device delivery that you see in green. How does this translate to patients? This is very important because we know that we need to shrink the tumors enough from critical structures so that surgeons can get to the tumor, take it out, and give patients a chance for cure.
Going forward, we have a clear regulatory and clinical pathway to approval. So based on the data that we showed you, we're really excited in going out and raising a Series A right now with the funds earmarked completion of our first in human safety trial, for enhancing our device to get ready for our phase 1B trial, and to expand our product pipeline. We've got a very strong team, and thank you for your time. I look forward to answering any questions during the Q&A session.